Good afternoon, ladies and gentlemen. I'm David Williams and I'm the Chairman of Inoviq. It's my pleasure to welcome you all here today to the 2025 Annual General Meeting. Mark's just told me we've got a quorum. It's 2 o'clock and I formally declare the meeting open. I'd like, before I start, to introduce my fellow directors. So right on the end of the table, stroking his chin, is Max Johnson. And sitting next to him with the new haircut is Val. And then coming back down the table, Skip Mary Harney, thank you, who's just come back from Namibia, alive, and she's obviously bought some clothes when she was over there. Only an African woman would wear that. But anyway, and then we've got Jeff Cumming, who's on a walking stick, who's just had his hip done, and Peter Gunsberg, who's just joined the board again, and more on that later. I'll get him to have a chat, but he's standing for election today. And then on the senior management team, I've got right next to you here, Leanne Inch. I've got the Chief Scientific Officer, Greg Rice, here. And I've got the CFO and Company Secretary, Mark Edwards, there. And we've got Dr. Emma Ball sitting here, who's not on the speaking agenda, but I may get her up later because she's the Chief Commercial Officer and she's been thinking for the last 6 months about business development and so forth. In the room, we've got Sudarshan from our lawyers, Minter Allison, one of the greatest corporate lawyers in Melbourne. That's what you told me. I've written it down, and he's done a lot of good things for Sudsy, but most importantly, kept us out of jail. And we've got Craig Stranger sitting over there on my left, right on my left, who runs Pack Partners, the brokers, and is also a shareholder now. Are you not? You are. People said you didn't have any money, but that's okay. I'm glad to see you on the register. I should say, not to be flippant about it, that Pack Partners helped us with the raising that we just did, and a bit more on that later. And no doubt after this event, he'll be telling us which analysts will be following the stock, and writing a report when that happened. Also in the room, we've got our auditors from Grant Thornton. We've got Michael Cunningham, who's retiring. Thank you. It's my sort of rotation. 5 years in. We've got Peter Glynn next to him, who's the audit partner taking over from Michael. And we've got Erin McLaughlin, more importantly, the person who does all the work. And they'll be here to take questions if anybody's got any regarding the accounts, if I can't answer them. So I'm going to go through the formal business quickly, which is just the motions and so forth. And then following that, I'll make a few brief comments. And then I propose to hand it over to Leanne, Greg, to make some comments, and then after they're done, I might come back and make some further comments before we wrap it up. And at that stage, depending on how we're going and how I feel, we might call Emma up as well, just to talk a little bit about partnerships and how we're going to drive BD and so forth. That'll be just a question and answer session. So you'll be provided the opportunity to ask any questions you like at the end of each item of business. But because of these hybrid meetings now, these have to be done and voted on as a poll, and that'll be done on all items of business. So when I come to each item, I'll put up on the screen how the votes have gone, but there'll be the possibility for people in the room, more particularly, how many people have we got online now? 24, but growing. Okay. They also have the ability to either change their vote or to vote by way of a poll, and so we won't know the final results until I close the poll, and Computershare will then collect any cards here and also look at the ones that are online, and so you won't get the results during this meeting, but we'll post them shortly thereafter to the ASX. So what am I going to do now? So I'm going to take the notice of meeting has been distributed to all registered shareholders, and I'm going to take it and the explanatory notes, the financial report, the director's report, the auditor's report as being read. The notice of meeting provides detailed information on each of the resolutions, but I'll allow you questions on any matters relevant to each of them as they come up. At the conclusion of the formal proceedings, representatives from Computershare will collect the voting cards if you have one, and the results of these will be put up on the ASX probably within an hour or two of this meeting finished. Okay, with respect to proxy votes, which will be shown on the screen soon, shareholders should be aware that the company has received proxies that representing approximately 33 million shares. Undirected proxies that have appointed me will be cast in favour of all resolutions, and the results of proxy voting for each resolution will be provided after each resolution is read to meeting attendees. Okay, let's go on then to the business of the meeting. The first item of business is to receive and consider the annual report of the company for the financial year ended 30 June 2025, including the financial report, declaration of directors report, and the REM report, and the audit. This item of business doesn't really require a resolution to be put to the meeting, but if there's any discussion about the accounts, this is the time to have it. So before I open the room, do you have any questions? No questions. And anything in the room? Nope. Good. As there's no questions or further questions of the auditor, Mike, maybe I should ask one. This is the last meeting for Christ's sake. You can go home, and so it was uneventful. But if there's no questions of the auditor, I'd like to thank Michael anyway. Seriously, he's had his 5-year rotation, which is required now by the Big Four, and he's off. But let's not forget that Erin was doing most of the work anyway. So you contributed a bit, Michael, but not that much. So resolution 1 then is the adoption of the remuneration report, and I'll put the motion that for the purposes of section 250R2 of the Corporations Act and for all other purposes, the remuneration report of the company and its controlled entities for the year ended 30 June 2025 is approved and adopted. Now, as you no doubt heard ad nauseum over the years, the REM report sets out our REM policy and provides details of our remuneration arrangements. The vote is advisory only and does not bind the directors, but it also comes with a voting prohibition. So in other words, we're voting on the REM report, and that means that a related party or a closely related party to me or another director doesn't get the vote. The results of the proxy voting you can see on the screen, and as you can see prior to the poll, which we're about to do, 79% in favour, against, and open, which I'll vote for those that are in my have been allocated to me. So in other words, probably 91% in favour. But don't take that as a reason not to ask questions. Does anybody have any questions on the REM report? On the screen? Nothing. In the room? Nothing. Thank goodness. All right. That is now, if you haven't voted already, vote on the poll. So I just watch my language that I'm not saying it's passed or not passed despite those things, because we still need to go through the poll procedure. Okay, resolution 2A involves the election of Dr. Jeff Cumming as non-executive director. And I'm going to read the motion that pursuant to, in accordance with Listing Rule 14.5, Article 6.3 of the Constitution, and for all other purposes, Dr. Jeff Cumming, director, retires and being eligible, is elected as a director on the terms and conditions in the explanatory memorandum. The results of the proxy voting are now on the screen, and we don't know who those 8 people are that voted against him, but we'll probably find them. Yep. So, but let's move on. Before I put the vote, I'd just like to ask Jeff to say a few words about himself. You might start off by just telling people why you've had a shoulder operation and now a hip operation, whether you're not beyond it, to be director. I mean, you're becoming a bionic man. I'm surprised you haven't got through security to get down.
Well, I didn't. That's why I was late.
Yeah.
No, I'm Jeff Cumming. I've been involved from the outset with Inoviq. My background, I'm historically a biochemist and a PhD in connective tissue biochemistry. Was managing director of Roche for the Oceania region, and as such had a lot of interactions with many of the people we're looking to do business with now, and have a fair understanding of what's required in order to take things through to the market successfully. And of course, in that sense, I'm just an adjunct to Leanne and Greg, because their background and knowledge is also very strong in the area. But that's a thumbnail to me, and thank you. I also recognise that we can't claim any success as yet on polling, but I thank you for your ongoing support.
That's very nice, Jeff. Thank you very much. And so I've already got a question from somebody that said, "Did Mary buy that title in Namibia?" And
yes, thank you very much, Mary.
Oh, my pleasure.
Thank you. Okay, I'll take any questions in relation to Jeff's re-election. Is there any questions online? Anything in the room? Nothing. Okay, I wish we were voting on your tie separately, because the voting would be a little bit different. I think, all right.
It's the man in the red jacket.
That's right, yes. Resolution 2B involves the election, I should say, of Mr. Peter Gunsberg as a non-executive director. And I'll read the motion that, pursuant to and in accordance with Listing Rule 14.4, Article 6.2C of the Constitution, and for all other purposes, Mr. Peter Gunsberg, having been nominated and consented in writing to his appointment and being eligible, is elected as director in the terms and conditions in the explanatory statement. Peter, you've been here before, but I'll ask you, first of all, just to make some introductory comments about yourself, and before I open it up for questions.
Thanks, David. I'm a finance guy by background in the most general senses. I did a commerce degree at University of Western Australia. Spent most of my working life as a stockbroker, and more recently I've been on the board of various companies over the journey. I've only got one other directorship in the Australian market, and that's as chairman of Metals X, which is Australia's only tin producer, and benefiting happily from a surging tin price. I've been a shareholder in Inoviq and its predecessors for many, many years. Inoviq, I don't know how many of you would appreciate, originally started out as a mining company, and it was the mining sector that my broking house specialised in. And I recently, obviously as a large shareholder, about 3.3 million shares, I'm very closely watching the progress and lack thereof the company has been pursuing. And on the back of the ovarian cancer results earlier this year, I convinced the Allied Group in Hong Kong to meet up with David and talk about the potential of them investing in the company, and with David's help, they've now done so. And I'm hopeful that with their money and a bit of good fortune and a lot of hard work, we can turn things around.
Thank you very much for that. So, yes, Seng Wee from the Allied Group had lunch with me, and over lunch I discovered that he owns a house 2 from mine in Double Bay. Mine was 2.5 million, his was 40 million, by the way. So he then invited my son down, who's got a separate tech business, and for breakfast, and he said, "You know what I like about your father?" And my son said, "What?" He said, "Because he's inappropriate." He didn't need to tell my son that. But anyways, I'm going to open this up for questions of Peter. Is there any questions for Peter on the screen? Nothing? Anything in the room? Nothing. Okay. Congratulations, Peter, subject to, of course, the voting on the poll. Resolution 3 is seeking the approval of a 10% placement capacity. This is a special resolution, which means under the Corporations Law that you need a 75% vote in favour. I'll read out the motion that, pursuant to in accordance with Listing Rule 7.1A and for all other purposes, shareholders approve the issue of equity securities up to 10% of the issued capital of the company at the time of the issue, calculated in accordance with the formula prescribed in Listing Rule 7.1A2 and on terms and conditions in the explanatory statement. Purpose of this is that under the ASX listing rules, a company has the right to make a placement up to 10% at any time without shareholder approval. But if you've already done it, then and you wanted to do it again, then you would need to go back to shareholders to get that approval. So this is really short-circuiting that, in a sense, freshening up our ability to do a 10% placement. So that's the purpose of it. The results of the voting on the screen. It's depressing, Jeff, because there's only 4 people against this, but there was 8 against you. So, is there any questions relating to the 10% placement? Nothing? On the screen? Nothing. Thank you. Resolution 4 then seeks ratification of the placement shares to institutional and sophisticated investors, which we've been talking about just then with Peter, and I'll read out the motion that pursuant to and in accordance with Listing Rule 7.4 and for all other purposes, shareholders approve the issue of 15.990.186 shares issued under Listing Rule 7.1 in accordance with the terms and conditions of the explanatory statement outlined in the notice of meeting. Peter's already given you a sort of a shorthand version about the Chinese, the people from Hong Kong, I should say, who put the monies in, and then the rest of the monies were facilitated by Craig and one of his colleagues in Hong Kong. I'll now take any questions, though, on resolution. Are there any on the screen? Nothing? Any in the room? Nothing? Good. Again, I'll just reiterate, if you haven't voted already and you want to change your vote or you're going to change it or vote on the poll, please vote now. All right, that's the closure of the formal proceedings of the AGM. I'd just like to make a few introductory comments before I introduce Jan and Greg. We're sitting here with a market cap of $50 million, and we're largely diagnostic, but with trying hard to move into therapeutics, and we will move into therapeutics. The highlights for me for the year, I'll come back, I'm going to make some comments at the end, but the highlights for me for the year is the fact that we had an ovarian cancer test that showed 100% success. There's a rather lengthy quote from me in the formal presentation, which we are about to come to. So that's number one, and that really sends us a good signal, and should send to the market a good signal about it being a very valuable screening test for ovarian cancer. The second highlight of the year, I think, is raising $10 million. So I don't know exactly what our bank account balance is today, but call it $13 or $14 million. So we're well funded to continue to develop our diagnostics program, but also continue to develop our therapeutics program. So now for us, it's as much about execution, about getting into therapeutics as it is anything else. More of that later, because I don't want to take Jan's thunder. But let me just introduce you to our CEO, and I'll come back and make some comments after that.
Thank you, everybody. I'm the CEO, Leanne Hinch, of the company. I've actually been CEO of the company now nearly 9 years, since late 2016. And the company started off as a single asset diagnostic company. We've obviously seen a lot of changes in this company during that time, and we've really refocused the business over the last couple of years under David's leadership to be an exosome company where we focus on our exosome research tools, diagnostics and therapeutics. Before we start the presentation today, I'd just like to make a few comments. I've noted that there's a lot of comments on Hot Copper about the recent capital raising. Everybody's aware that a number of our directors participated in that capital raising. They want to know whether the CEO put her hands in her pocket and participated in the capital raising, and I'm certainly pleased to say that I did, and I have participated in every capital raising for the last, I think, 3 or 4 years. Furthermore, I have bought shares both in capital raisings and on market, and own about 620,000 shares. So I have participated, backed this company and backed our science, as do our entire management team. I'm certainly pleased to say that every one of my management team has invested in this company and continues to invest in this company. So we all back this company in science, and we believe in the future success of our pipeline. The other thing that I want to point out is people are asking about our partnering program and that there's not been a lot of progress. Partnering is an ongoing process. We actually have numerous conversations going on with companies that are both clinical laboratory companies as well as major diagnostic companies. This is about both our development strategy and our commercial strategy. Important to these conversations is actually about informing the development strategy of our exosome ovarian cancer test so that it is ready to be commercialised as an LDT by the end of December 26. So those conversations have actually informed the development program and will be important to ensuring that we meet that milestone and sign a partnership deal with a clinical diagnostics company so that it will actually be in market earning revenues for the company in 2027. So if we consider that one of our highest priorities for the company. The other thing that we're asked about is our sub-B2M program. As I mentioned, really in the last couple of years, we have focused this company because we've got to focus, we've got to prioritise, and we have to resource the company accordingly, including with the right people, such as the appointment of Dr. Emma Ball as our Chief Commercial Officer, in order to take our programs forward. So we really have focused on our exosome program. In order to further commercialise and partner our CA15-3 breast cancer monitoring test, we need to transfer that onto a high-throughput instrument platform. We need to do that before we can actually partner and commercialise it. So that's the second priority to our exosome program, and it's really for a clear reason. It's probably about 2% the size of the ovarian cancer market. So the focus of our funds and management's time is really on taking that program forward. Lastly, the CAR exosome program for our therapeutics, we consider that to be a very important part of progressing this company. What we've really done is taken our technology, we've put it in a box, we've sold it into a research tool market that's worth about $500 million overall. So that includes everything, whether it's instruments, all sorts of other reagents, and we're just a small part of that. Effectively, by commercialising that technology, which actually underpins our exosome ovarian cancer test, that market's 10 times the research tool market at $5 billion. Investing in therapeutics, which is where the company's intellectual property is, where our capabilities and expertise are, makes that another 10 times market size, now looking at a $50 billion market. And that's just for one market alone, looking at breast cancer. So that's why we've invested the company's time, shareholder funds into moving from diagnostics into therapeutics. So with that as a start, I'm now going to focus today's presentation on our exosome programs. So hopefully that's answered some of the questions up front. Greg, perhaps you want to jump up as well for the presentation.
That's an invitation, Greg.
Yeah, it was.
No, he's getting a bit old. He's hearing.
I was wondering. Greg, did you want to say what?
I'll come up later.
You'll come up later.
Yeah. Tag team.
Right. So everyone's seen this slide before. It's pretty much the opening of every presentation we do. But really the point is that we are a next generation diagnostics and therapeutics company. We are focused on exosome-based products, and they are for cancer. We have a patent-protected exosome technology platform. It's got multiple applications across research, diagnostics, and therapeutics. This enables us to have multiple opportunities in terms of milestones coming up over the next 12 and 24 months. Our exosome research tools are already commercialised. They're available in market through our distribution partner, Promega Corporation, who's actually selling those products throughout the globe to academia, to diagnostics companies, and to therapeutics companies. And what we've seen with Promega over recent times is that a lot of the sales are now moving over to the larger diagnostic and therapeutic companies, which are really important to the company and also resulting in ongoing collaborations for us at developing custom exonet tools. We have a number of assets in the exosome portfolio. So that includes our priority program, which is our exosome ovarian cancer screening test. So that is our priority. We are focused on getting that to market as an LDT by December next year. So we'll intend partnering that with a laboratory company before then, obviously. The other area of priority is advancing our preclinical stage exosome therapeutic, which is initially in development for triple-negative breast cancer. But clearly, given that we have an ovarian cancer screening test and the company's strategy is to be an integrated exosome diagnostics and therapeutics company, we do intend to expand that pipeline into ovarian cancer. We will be partnering in order to commercialise these technologies. We are a small company. We don't have the funds and the global footprint to commercialise these technologies globally, so we'll be doing that with partners. And we do have an experienced leadership team and board in place with an advisory board, backed by an advisory board that is world-class in terms of its exosome science, its cell therapy development, its diagnostics development and commercialisation in these global markets. As you're all aware, we recently completed a $10.2 million capital raise. That resulted in us having a pro forma cash balance of around $14.3 million. Also in November, we got an R&D tax refund of $1.3 million. Just on that, just pointing out that sometimes we're asked, why do we only get a tax refund of $1.3 million? It's because quite a bit of our research is actually done in the US and we can't necessarily claim all of that back. So I just wanted to point that out. Management and the board owns about 8% of our shares. As I pointed out, the majority of the board and all of management own shares in the company. So what's our strategy? How are we driving growth and value into our business? It's really the exosome technology platform is what underpins our entire portfolio of research tools, diagnostics and therapeutics. So our research tools is really our technology in a box solution that we've partnered with Mega Corporation. We are selling around the globe. We've got initial revenues. They are being reinvested and will continue to be reinvested as they grow into diagnostics and therapeutics. Importantly, we see that the real revenue from our excellent research tools will come from collaborations that we have with diagnostics and therapeutic companies that are using our tools, and particularly custom versions that we develop for them, for potential diagnostic products, as well as for what we call target discovery for potential therapeutics. So we see that as an important component of our portfolio, and it does actually underpin our diagnostics. We wouldn't have an exosome ovarian cancer test without ExoNet, which is our research tool. So that enables us to capture our exosomes. We've developed our exosome ovarian cancer tests to the point now that we've shown we can detect 100% of early stage disease with minimal false positives. We got actually over 99.6% specificity, which is really important for a screening test, and Greg's going to explain that to you later. On the therapeutic side, as I said, we really have an integrated diagnostics and therapeutic strategy. We're focused on developing high-value therapeutics because the return on investment is so large in these markets. The company's really seeking to produce a 10-time return on investment as we grow from research tools to diagnostics to therapeutics. Each market's 10 times. I'm going to hand over to Greg now, who is going to come up after that. Yep. And he's going to talk to you about why our XNet technology is superior and how it underpins our portfolio. Right.
Hi, everyone. I get the easy bit to tell you a little bit about what exosomes are. I think the easiest way to think about what they are, they're small packages that contain biomolecules, and they're released by cells to communicate to other cells. So they're packages that contain information, communication to other cells. These exosomes are released from cells and they travel through the body fluids, including blood, to the target cells. Once they arrive at the target cells, the target cells internalize them, and those messages that the exosomes carry actually change the function of those target cells. So, for example, here, if it's a cancer cell that's sending out these exosomes to a normal cell, it might be telling that normal cell to allow me to grow, to proliferate, increase my blood supply so I can survive. So this was their function. In terms of our diagnostic program, we can leverage, we can tack into that communication. And what we've done is to develop technologies that capture exosomes that are, for example, released from cancer cells. We're able to read those messages, to quantify them, and therefore build better diagnostics, more specific, because we know they're coming from cancer cells, more sensitive, and we can develop earlier detection of these types of diseases. So that's very simply what exosomes are, how they fit into our diagnostic program. You might ask, well, hang on a sec, Greg. All cells release exosomes, and that ends up in the blood. Isn't that going to get a bit confusing? That's a very good question. And it's quite right that all cells release exosomes. They carry different messages. They target different cells. Ooh. However,
Thanks.
all of those exosomes, thank you. Okay. All of those exosomes are pretty much the same size, with the same density. They carry the same electrical charge on their surface. And they have some common exosome proteins called tetraspanins that sit on their surface. Isn't that going to get confusing? And yes, it is. They all end up in the blood, and you have this heterogeneous mixture of exosomes sitting there. And this has been one of the major challenges in trying to develop exosome diagnostics. Our competitors in this market space use bulk isolation methods that are dependent upon the physicochemical properties of exosomes: size, charge, density, and those common tetraspanin proteins. And what happens is that they all end up in the same bucket. And it's very difficult to identify the actual signal coming from the cancer cell. So they have a low signal to noise ratio. Yes. What we've done is to develop a technology where we can specifically pull out subpopulations of those exosomes. So we can pull out and enrich those ones that come from, for example, ovarian cancer, and put them into a bucket that we can analyse. So what we end up with is being able to separate the important exosomes, the one that tell us information about ovarian cancer, for example, and increase the signal-to-noise ratio. That allows us to develop more sensitive, more specific diagnostics, and allows us to detect the cancers earlier. So that's how exosomes fit into our diagnostic program. But there's more. We can actually flip that communication system around for our therapeutics program and use it to talk directly to cancer cells. So in relation to our therapeutics program, what we've done is we've developed a technology that allows us to instruct immune cells, such as natural killer cells or T cells, to make exosomes that specifically target cancer cells, for example, ovarian cancer or breast cancer, and send them a message to die. And it's that simple. So what we've done here is that in the lab, we educate these immune cells to produce those exosomes, that we can make them to target breast cancer or ovarian cancer. We have developed a technology to isolate those exosomes for therapeutic use, and then we can put them into either cells on the bench, into animal models, and then ultimately into humans. So it's that simple. We've just harnessed the communication between cells, we use it for diagnostics, and also then to convert it into therapeutics.
Right, now I'll answer the money question. So everybody wants to know, how's the commercial progress going with Exonet? As I said, our global distribution partner is Promega Corporation. They have been delivering initial revenues under their early access program for the last, I think it's about 18 months to 2 years now. And that's really building traction, if you like, ahead of their full commercial launch, where they're actually launching our product combined with their products, because they specialise in a whole range of research tools, but one of their important areas is RNA extraction. Inside exosomes is RNA. They've determined that a really important market is RNA from exosomes. So what they've done is they've created combination kits that uses our ExoNet exosome isolation technology, as well as their RNA technology, and they're running it on their instrument platform called Maxwell. That platform they have in the majority of their academic and diagnostic customers that they actually have. And importantly, that will enable them to increase sales. You can see that the graph on the right, do I have a content? Don't know. The graph on the right, yep. We've gone from, pretty much 0 customers back in first quarter of calendar year 24, so say January 24, to now we're sitting at 70 customers at the end of October with Promega. We think that customer growth and certainly the revenue growth will increase in first quarter of next year, when we expect them to launch their fully automated ExoNet combined with RNA solutions to their customer base, which they can roll out to customers that already have Maxwell. They obviously see that as an opportunity to sell Maxwell as well, but that's good for us as well. Importantly, we have expanded the exosome or the ExoNet portfolio with NeuroNet. We've spoken about that before. That's for capturing exosomes that are derived from the brain. We also have TexoNet, which is about the exosomes that are derived specifically from tumours or cancer. And then importantly, custom nets. What are they? They're actually specific nets that we develop for diagnostic and therapeutic companies for their own purposes. Basically, many companies are looking at specific diseases, whether that's cancer, also called oncology, neurology to do with the brain, cardiac disease, etc. What they want is a custom solution, as Greg pointed out earlier. It's about getting these specific subpopulations of exosomes, which enables earlier and accurate detection of disease. So they actually pay us to develop these custom nets, and ultimately, if that's successful, they'll license those nets from us for use in their final products. So they're really important relationships to us. They're increasing. We have a number of feasibilities that are ongoing at the moment. As they expand and get to a point that they're announceable, then we would expect to announce them to market. Additionally, I've already pointed out that we're paid for it, so that's the service agreements where we are undertaking this biomarker discovery and diagnostics development with these parties. And really, I guess the end point of this whole slide is that it is about getting the customer and revenue growth, which will be driven by both Promega sales of ExoNet, standalone and in combination, as well as by our future license revenues from our partners. So one thing just to point out in terms of the graphs, we are seeing increasing sales in Europe. That is probably driven by the fact there is reduced spending in the US at the moment within the academic segment, with the current administration reducing funding to academia and research institutions. So Europe really has taken over. The other is when we look at the customer type, when we first launched this product, it was pretty much all to academia. Reason being is we were seeking publications with research groups on their use of ExoNet, so people learnt about ExoNet. So the combination of what we did there, together with Promega marketing the product, has led to a lot of the collaborations we have today. So we're now seeing a lot more pharma biotech companies starting to come either direct to Promega or to us in actual fact for collaborations, as well as clinical and hospitals that are, or I should say clinical labs and hospitals that are developing their own exosome-based products. So I'm going to start with our ovarian cancer test and how we're planning to commercialise it. Effectively, we have a dual path commercialisation strategy. Now, what does that really mean and why are we doing it? We're doing it because the idea is you can commercialise a test first in the United States as what's called a laboratory developed test, or LDT, and later you can then fully commercialise it both in the US and globally as an in vitro diagnostic kit. That enables us to maximise speed, access for patients, as well as value to shareholders, our clinicians, and the health economic system overall. So when we look at the LDT strategy, why are we going as an LDT in the US? It's because it's the fastest path to market. It enables early access of our product to patients. It enables early revenues for the company. And importantly, it enables us to build real-world data which we can use to support our in vitro diagnostic future filings. The difference between an LDT and an IVD is we have to commercialise the LDT with a single laboratory partner. We can't sell it to multiple labs. We can only sell it to one lab. What does our partner look like? It's a Sonic Healthcare reference lab, or it's a LabCorp central lab, or it's a Quest lab. They're the sorts of partners that we're looking at, or it could be a specialty clinical lab that specialises in women's health. We expect, as I said, this product will be at market, or will be ready to be commercialised with our partner by December of next year, so 2026, such that we'll be earning first revenues when it gets launched in 2027 by our partner. How do we ensure that it's adopted? And again, I read on Hot Cuppa, everyone wonders why we keep putting publications out there. You can't commercialise a test and get clinical adoption without having publications. Publications in the world of science is everything. And I think that our chairman can attest to that, because that's really what he did with PolyNovo, is made sure that there's loads of publications out there by clinicians. We're doing the same ourselves, and ensuring that our advisory board is helping us to present that data together with our Chief Scientific Officer, Professor Greg Rice, at clinical conferences around the world. We did our first conference, as everybody probably knows, at ASCO, which is one of the leading clinical conferences in the world for oncology. We'll continue to put new publications out and build our KOL network coming up. From a reimbursement perspective, i.e. how do we get paid? Initially, it will be patient pay. It usually takes 12 months to 24 months until you get reimbursement. Yes, you can get reimbursement as an LDT, but usually the figure's larger as an IVD. So once we do get this on market, we'll continue to develop the product. Ultimately, what we're seeking to do is to partner this with a large diagnostics partner. That's Roche and Abbott, Hologic, Danaher, those types of companies. Where our focus is on getting this to market as an IVT 2029 onwards, and that really results in much larger revenues because it enables global market reach, where we look at the US, the UK, Europe, China. Everybody knows that our cornerstone investor was a Chinese-based or Hong Kong-based investor who's got hospitals in China. What they do is they actually deliver what's called diagnostic screening programs to corporates, and hence their interest in the exosome ovarian cancer test is to really get that into their hospitals so that they can screen patients in China. Importantly, what do we have to do in this case to get adoption? We need to complete a pivotal clinical trial. Once we've done that, there'll be various health economic modelling and to help us put that into clinical guidelines, get reimbursement at the best rate we can, which we expect to be at very reasonable levels for this type of test, and ensure that we've got both public and private payers reimbursing the test. That enables us to get broad clinical adoption of the test. Basically, clinical adoption is driven by this inclusion in clinical guidelines. Patients buying the test, we want to see reimbursement by the payers, and that enables sustainable growth for the company from this product. And I'm now going to hand over to Greg to tell you why ovarian cancer, or what an ovarian cancer screening test actually is, and what the performance requirements are.
Thanks, Leanne. So, just a really brief update on the ovarian cancer screening test. And I think it's really important to understand a few things about where the test sits in the market, how is it different from an early detection test. What we're doing is building an ovarian cancer screening test. So that's for use in asymptomatic average risk women. So something like the breast cancer screening program. We're targeting women of 40 to 74 years old. And these are women that have no symptoms. They're not at high risk. And what we're trying to do here is to identify the cancer early so we can actually do something meaningful about it. That differs from an early detection test. Early detection means the finding or diagnosis, for example, of ovarian cancer at an early stage. And this almost exclusively relates to women who have a high risk of developing breast cancer or who have symptoms. So these are the patients with BRCA1, BRCA2 Lynch syndrome or a family history of breast and ovarian cancer, and who have these symptoms and these risk factors. So it's not a screening test. It's never going to be able to be put into a general population for screening, and I'll show you why. It's simply for trying to identify women at risk who have developed ovarian cancer. Screening test is completely different. What we're doing here is regular testing of women in the general population. They don't have symptoms, and what we're trying to do is to catch the disease early when we can actually do something about it and improve mortality rates. And for that reason, the criteria that you need for a screening test in the general population is a sensitivity of greater than 75%, i.e. you get identify at least 75% of the cancers that the test is run on. But more importantly, relates to the specificity. You need to be able to tell women that they do not have ovarian cancer when they don't. So you need to be able to clearly identify those women in the test who do not have ovarian cancer. You don't want to be telling women you have ovarian cancer when you do not. The graph on the right illustrates this for the Australian market. On the y-axis, we have the number of women who are told they have ovarian cancer when they do not. On the x-axis, we have the specificity of the test. So if you have a test that has a specificity of 94%, what that means is that you're telling over 200,000 women each year they have ovarian cancer when they don't. And that has significant consequences, both for the women in terms of the follow-up, so imaging, surgery, but also for the health economics of the country. In terms of looking at a test with a specificity of 99.6 or 99.9%, you're only telling less than 3,500 women that they have cancer when they don't. This is the difference between a test that perhaps looks like it's performing well at 94%, but it's not, and a test that is performing well and reducing the number of false positives. So that's important to understand. And there are a number of tests out on the market that are looking at early detection of ovarian cancer. They can be applied to high-risk populations, but they're not going to be applied to general population. In terms of our XOOC test, this was the latest data that we obtained in the OC 500 study. And what you can see here is that we were able to, in this cohort, using this algorithm, identify all those women correctly who did not have cancer, all those women who had stage 1 and 2 disease, cancer, with an overall sensitivity of 77.4%. So this exceeds the recommended requirements for a population screening test. Where are we going with this? What's the next step? In terms of what we need to do, we've had discussions with some of the major pathology providers in the US, and they've really informed us about what we need to do to be able to implement this test as an LDT, for example. And the types of clinical studies that we need to do is, 1, do a larger cohort study, 2, look at diseases that might confound the test. So diseases such as diabetes, endometriosis, breast cancer, etc. And also demonstrate that it works on the high-risk group. So, as I say, these have been informed by potential partners in the US, and this is what they're looking for. We have about probably 90% of those samples collected, and we'll start to assay those next month. In terms of moving this into an LDT in a clear laboratory, What we've planned here is to do an analytical and clinical validation studies in those in the CLIA lab on the LDT pathway, as Leanne has mentioned. And in parallel, we've actually started to develop a better understanding of the pathway for IVD development, seeking to put in a breakthrough device designation submission, leading on to a clinical pivotal study in 27 and going through regulatory approval. So this is what we have planned for this type of development process. Over to you.
All right. I'm just going to do a quick introduction on our CAR exosome program, which is really about developing a next generation cell-free therapy for solid tumours. So everybody knows out there that effectively CAR T or autologous CAR T therapy has been enormously successful for treating blood cancers. To date, the problem is it's very, very expensive. It costs about $500,000 to dose a patient, and it's been successful, as I said, blood cancer, but not for solid tumours. There's many companies developing alternative cell therapies. What we're developing is what we call a next generation cell-free therapy. So we're relying on the exosomes produced from those cells, as Greg explained earlier, in order to deliver the same impact in terms of targeting and destroying solid tumours. So effectively, this is a platform technology. It's got multiple applications. Our focus is on what we call our CAR exosome therapy, but it can be used, and it's something we may look at down the track, for drug delivery, regenerative medicine, and aesthetic medicine. In fact, there's a massive aesthetic medicine where women are putting exosomes on their face for enormous sums of money. It's a multi-billion-dollar market, but women do put many things on their face. So, so in terms of our focus, it is, as I said, on solid tumours. Our first program is triple-negative breast cancer. I did allude to earlier that given we have an integrated diagnostics and therapeutic strategy, we do intend to expand that into ovarian cancer, and Greg will talk briefly to that in a moment. Why are we doing it? It's because our CAR exosomes inherit the same tumour targeting specificity as their parent, either T cell or NK cell. The important thing is that because we're actually taking them from a cell line and we're not taking them out of an individual and putting the cells back into an individual, ours is an off-the-shelf therapy. So we're not using cells. We eliminate a lot of the safety issues related to immune rejection, cytokine release storm, etc. And we massively reduce the cost of goods because we're not doing this manipulation from a human and putting it back into a human. And when we talk about the cost of goods, we really compare it to antibody therapy, which is a well-known pharmaceutical model for delivering drugs to patients. In this case, the patient doesn't have to wait for their therapy to be developed and put back into them. The drug's sitting on the shelf waiting for them. It's called an off-the-shelf therapy. That costs about $165,000 as an average cost to deliver to a patient versus CAR T therapy that's about $500,000. And you can only use it once. So this is over a period of a year, multi-dosing. That's how we expect our product to be delivered, with huge benefits in terms of potential efficacy for solid tumours, safety, and lower cost. And I think I'm handing back to Greg to talk about upcoming studies.
Okay, so yeah, look, this has been a really exciting program, and it relates to my earlier comments about flipping that communication between cells around. And it gives us the opportunity to actually talk to cancer cells and tell them to die. What we've done here is to use those natural killer cells and T cells and instruct them to make these specific exosomes that kill triple-negative breast cancer cells. So the early start was to generate a master bank. This is a bank of immortalized cells that we can draw upon. We've instructed them. The instruction is a CAR, a chimeric antigen receptor. It pops it onto the surface of the exosome, and it goes away and finds where it can bind. And where it can bind is a triple-negative breast cancer cell. We have also developed technology to purify those exosomes for therapeutic use. So this is our ExoAce technology. So we can actually produce these exosomes in therapeutic dose quantities. We then went on to look at proof of concept studies. And we did these in our own laboratories initially, where we looked at these educated T cells and these educated natural killer cells. We grew them up in the lab and we put them on cells in the lab to see whether they worked. And this is some of the results that we obtained. We've shown this before, but essentially this is live cell growth on this axis, the y-axis, time on the x-axis. This line here is the untreated cells. So this is how the triple-negative breast cancer cells just grow in culture. Here, we've treated kidney cells with the same instructions, and they don't do anything. Here, we've educated the NK cells to target triple-negative breast cancer cells, and they kill around 88% of those cells in 96 hours. So really strong positive data that we do have something here.
And we might just mention that that data was validated by Peter Mac not long after.
It's not to say that. Those studies are done in-house, and clearly you want a third-party independent validation. And who better to do it with other than the Peter Mac? And what they found was the same. Actually, they found markedly better results than what we did. So again, it's really strong, positive proof of concept data. What do we do next? Obviously, we wanted to try this in an animal model. So we chose a mouse model. We gave the mice triple-negative breast cancer. This was done by a CRO, so it's independent of us. That study is pretty much complete now. We're waiting on just some data to come in, looking quite positive, and we'll release that data in December. But based on that's both the lab-based studies and also the animal studies, what we've planned now is to make our own immortalized T and NK cells. You want them immortalized so you can continually grow them. You don't have to go back to patients to get samples. So we've embarked on that process. We've planned preclinical studies, both in breast cancer and ovarian cancer for next year, and also starting to commence the IND enabling studies that we need to make a submission ultimately. The next steps after that are really identifying a CRO in the States and the sites where we're going to do these types of clinical trials, get approval, which is called Study May Proceed from the FDA, a bit of a mouthful, and site activation in 27, which will lead on into the first patient trials. So a very promising program. I think in terms of therapeutics, we've actually progressed it really quite rapidly, and some really strong positive results. I think over to you.
I guess I might just finish off on the therapeutics program. So what have we got to look forward to? As Greg just pointed out, our in vivo efficacy data, in fact, it's safety, efficacy, and biodistribution data, we will be delivering in December of this year, as per our milestones. And importantly, in the next half of next year, we'll be progressing our manufacturing, which is all about our future clinical trials, and doing these preclinical studies in both triple-negative breast cancer and ovarian cancer. And it's all about the goal of commencing these IND-enabling studies, which enables us to dose our first human, which for obviously breast or ovarian cancer will be females. Now, we expect that to be 2028, just putting a timeline out there. That is the focus of the company with this program. That's actually a very fast program. As Greg pointed out, we've had really good success with this exosome program. We expect it to be successful. We're very excited by it as a company. And that's really the company's capabilities are all in that exosome sites. The exosome ovarian cancer test, we've already commenced our clinical studies, as Greg pointed out, for ovarian cancer screening. As we already said, informed by our partner discussions or potential partner discussions, and they're underway. When we'll start analysing some of those samples that have come in I'm going to say next year. Greg said next month, but I'll say next year. So we will expect to report on that in the first half of next year, with a focus really then on strategic partnering for LDT commercialisation. That's as per the milestones that we've put out there before. That's already underway. We've been, obviously, these are ongoing conversations. And importantly, we also are progressing in parallel our IVD clinical and regulatory strategy. Again, they're informed by the conversations with these same parties. So we will, I pointed out, put clinical data out there in that first half of next year, followed by analytical and clinical validation in a partner lab. And then the test will be ready and partnered to be commercialised in 2027. So we've got a lot coming up. That's really the focus. ExoNet, we do expect sales growth, increasing collaborations and diagnostic partnering out of that product, which will lead to further exosome-based diagnostics in the future. So from my perspective, the company's in a really good position. We are looking forward to those milestones being reflected in our share price coming up in next year. But effectively, there's a lot going on with collaborations with our research tools, our exosome ovarian cancer test being really ready for commercialisation by December next year, and a lot of results coming up with our preclinical program for CAR exosomes, which we consider very, very exciting. So I think we're handing back now to yourself.
Before you sit down, are there any questions from the floor? Leanne? On the screen?
We do have a couple of questions.
Oh, sure.
A couple were probably well addressed early on in Leanne's presentation, where she spoke about the management shareholding and the director shareholding and how they participated in recent ways. So we won't go into that in any further detail. Another question was around what does management have to plan to increase the share price of $1 by July 26th. I think what Leanne just spoke to then around the future catalyst addresses that on the slideshow. Question from David Norris around the large shareholder that has been selling, where they're at, and has that ended? My view on that is I think the Biotech Capital Management Fund has sold out. They were the major seller during the year. So I'm not sure.
Are they completely out?
They are out now, yeah. So I'm not sure if you have anything to add on that front.
No.
I think the only we saw some selling after our recent capital raise from a few new holders. I think that's quite common when you do an initial capital raise. Importantly for the company is we needed to fund the company to take these programs forward to hit these big milestones, which will grow our share price. So that's a short-term hiccup that we're sure will be correct.
Question again from David Norris around plans for any further capital raises whilst the share price is at record lows. No, that's very clear. Progress on the XOC validation, 2,000 samples. It's on track and planned for the end of this quarter.
Yep, I think we've spoken to that at length.
Yep, getting the samples underway. And that's largely it. There's sort of a question around the commercial strategy, which was covered off as well, the partnering slide. And a question from Alex Last. Can you tell us what kind of revenue is Promega likely to generate in FY26?
Well, look, we're not in the business of giving forecasts. It's really just started. We've got 70 customers there, and they're changing their model. So we'll suck it and see. Putting our neck in that news just yet.
Okay, that's the questions online at the moment.
All right, great. Thank you. Look, before I... sorry, are there any questions from the floor? Yeah? No? Nothing? All right, good. I might just take a few minutes to get you up, Emma. This is Dr. Emma Ball, by the way. Are you a doctor?
Hey, molecular biologist.
Oh, you are?
Many moons ago.
Thank you. Stand here so people can see and they don't see my gut. So, what's your position?
Chief Commercial Officer.
What does that really mean for our shareholders?
It means partnering, a lot of work around, so my main focus is partnering the XOC test, as Leanne's mentioned. So we're in multiple discussions with multiple partners on that. So that's part of it. I was also closely involved when we licensed in the technology from University of Queensland. So it's all about those who are doing those pre-commercial deals, as well as making sure that our commercial partners in terms of Promega are delivering, and we're supporting them to do that.
That's great. So you're in your helicopter, you're looking at what we're doing in Bavarian.
Yep.
How are you sort of thinking about that? Are you thinking about that as being just a pathway to LDT, the laboratory developed test, or you have a sort of way of thinking about it where it's in different silos? That some of them are pathology labs, some of them are.
Absolutely.
How do you
And
sort of
actually,
think?
do we have that diagnostics deals slide? We do. I can. No, okay, that's right. I can talk to that. So, as Leanne pointed out, we're pursuing a two parallel process, one in terms of laboratory developed tests, and then also as an IVD. And I think what that was it. Yep. There have been a number, so 3 in particular, really landmark deals that have happened recently that I think really validate the liquid biopsy cancer screening space, but more specifically the approach that we're taking in terms of the LDT IVD parallel pathway. So the deals that I'm talking about are the top 3 here. So Freenome is a liquid biopsy company, centralised laboratory in the US. Exact Sciences, pretty similar as well, also centralised laboratory in the US, also doing liquid biopsy, predominantly LDT. Exact licensed Freenome's test, colorectal cancer test, for $885 million, and that's only for the US. Leading Freenome to also continue to develop it as an LDT.
So before you go on, those sort of deals you're talking about, they're just buyouts, right? So
no, that's a licensing deal.
Okay, so license.
So that's a good analogue for what we're aiming for. The next one, I think, shows something different. The next 2 show something different. So again, Freenome, centralised laboratory, LDTs predominantly. Roche have now licensed that technology to develop distributed kits. So that's the IVD, that's the LDT IVD parallel pathway. So $200 million, and they've also invested equity. I think the other huge landmark deal was announced this week, and that was Abbott, again, as the decentralised diagnostics leader, acquiring Exact Sciences. So again, that LDT model. So for $23 billion US. So I think that really validates exactly what we're trying to do here. It validates the space that we're working in and the commercial strategy that we're applying.
Yeah. So when Leanne talked briefly about Sonic and a number of other what I'll call path labs or something anyway, but what do you expect for something? Do you think this is the leading candidates that are going to be interested in us at path type level, or
look, it could be either, as we've seen here. It could be a centralised, say, a Freenome or an Exact Sciences type company, or it could be a Roche Abbott that have more that distributed, and maybe, it's one for the LDT and it's another for the IVD.
Okay. Is there anything else you want to touch on?
I think that was the deals. I guess the other thing is in terms of ExoNet. So our focus is a lot around the XOOC and partnering that, but then ExoNet as well. We're starting to see through our relationship with Promega, they're selling it for research use only. That's actually now starting to drive potential partners to us for clinical commercial use, where we can develop custom nets or NeuroNet, other nets for those companies. So that will ultimately result in licensing income for us.
Leanne also talked about the importance of publications.
Yes,
so
absolutely.
My view is if you're selling it into research houses, that ought to lead to patents.
Absolutely, yep.
And are you seeing any sort of cracks in that?
Yeah, we are. And I think the importance of some of these partnerships is that it mainstreams our technology as well. It validates and mainstreams that technology. It gets the story out there, and hopefully then brings in other potential partners. And also, as Leanne said, it's important for, say, the reimbursement piece, making sure that we're getting on clinical guidelines once diagnostic tests are developed.
All right, that's great.
Great.
I'm not going to allow questions to be asked.
Thanks, David.
Take a lot of time. I just wanted to introduce people online and in the room to Emma anyway. Just get a feel for how they're thinking about BD, because the partnerships in there is going to be really an important part of it. That's the end of the meeting. I'm going to close the voting now for the poll. So if you'd like to collect any cards people have got, if you haven't marked your card already, please do. Just wait till they're all collected. Nothing? Good. Thank you. Get to work. All right. Thank you, everybody, for coming. And that's the end of the meeting. And hopefully it looks like it's going to be an exciting 12 months.
We certainly think so.
9 months left. Thank you all for coming.